Thanks everybody for coming. I'm really excited to be able to host and introduce Alex Nechiporuk for the Genetics Colloquium. Alex started at OHSU, Oregon Health and Sciences University in Portland, Oregon in 2008 as an assistant professor. And I just learned was recently promoted to full professor. So congratulations, Alex. His lab specializes in using zebrafish to study questions of developmental and cell biology. And largely they've focused historically on the lateral line sensory system, both the afferent sensory axons and neurons, as well as the sensory organs that make up the system, as a good model for neuronal cell biology, as well as collective cell migration. So they have used the system to address fundamental mechanisms of growth factor and morphogen regulation of proliferation and migration of the cells that develop into the sensory structures of the lateral line, as well as the cell biology of neurons, both axon structure and function, using the system. And they've used a variety of techniques, including classic forward and reverse genetic screens, and really beautiful in vivo imaging, which you can see here. And these works have been published in a variety of really great journals, including Science, PLOS Genetics, Development, JNeurosci, et cetera. So Alex and his lab have done an amazing amount of diverse work during his time at OHSU. And what I'm most amazed by really is how readily they accept technical challenges to better their science. So Alex is not one to let techniques stand in his way. There's a question worth answering, they'll figure out a way to do it. And that's admirable. So this is really well exemplified in the work he'll tell us about today, where they've used single cell RNA sequencing to understand the unique biology of pioneer neurons in the lateral line system. This subtype of neuron is largely unexplored actually, and is really essential in both the peripheral and central nervous systems as they set up scaffolds for axon outgrowth at later developmental time points. And we don't really know nearly enough about them, so I'm excited to hear what his lab has discovered about this really unique set of neurons. Alex.
Thank you, Katie. Thank you, everyone. You actually described work in my lab way better than I would have, so I really appreciate it. And I also appreciate people spending time today with me, sharing their data, and students talking to me. So it's so nice to be out and actually talking to live people. As Katie mentioned, what I'm going to do today is actually try to convince you that this type of neurons that is found throughout the developing nervous system called pioneer neurons have actually distinct molecular makeup compared to the close relative population called follower neurons. And again, as Katie mentioned, we used a lateral line system to actually study neurogenesis and cell migration. And lateral line system is a mechanosensory system of aquatic vertebrates that senses water movements and controls various swimming behaviors. And it consists of two main components: the sensory organs themselves and sensory nerves that innervate those sensory organs on the surface of the fish trunk. And during development, basically, what happens that this group of cells shown here in magenta called posterior lateral line primordium will migrate along the flank of the zebrafish and will deposit clusters of cells which will differentiate into sensory organs. While pioneer axon terminals are embedded within this population of cells, there are typically 3 to 6 of these pioneer axons, and I marked the growth cones, which are just bulges with arrowheads. So they're guided by this group of cells toward the very tail of the embryo. So the work that I'm going to tell you is actually all done by graduate students. So it's Ben is leading the project with the help of another graduate student in the lab, Lauren, and Nick was indisposable bioinformatics help in this project. He is in Laura Heiser's lab. So what are the pioneer neurons? And you probably heard this name many times in different contexts, but its name implies they're the first to explore the environment and first to extend during development. Essentially, they're like little explorers. They go into unknown and guide other axons called follower axons to their target. Okay, so they encountered in peripheral and central nervous system. They found in invertebrates and all types of vertebrates in very many different contexts. And at least in some or many contexts, they are known to have more elaborate growth cones. And it's not surprising because they are responsible for this initial target pathfinding behavior. And at least in some contexts, they may also have larger cell bodies. And in some cases, they even innervate distinct targets at the end. So the way I like to think about pioneers is that they actually set up initial scaffold of the developing embryo, which then other neurons like follower neurons will use to navigate to their target. So it could give you an example. So this is in 8.5 weeks post-gestation human embryo that's stained with antibody that recognize neurons and their axons. And you can see that it's actually only a few millimeters big, but most of the CNS and PNS tracks already laid out. And then, this embryo will grow more than 200 times, okay, if you compare to an adult. However, there is very little of this de novo exploration after these stages. So what we're talking about for most part is sort of filling in and growth of these axons. And of course, this is not to scale. If I scale this embryo, it would be like a dot on the screen. Okay. Now, one interesting question, and I'm kind of relative newcomer to, subfield of pioneer neurons, is to ask, are they actually required for finding targets? In other words, if you remove pioneer neurons, either ablating them or genetically, what happens to the follower neurons? Can they actually find a target? This is an important question because if they are distinct, it would imply that they have a different molecular makeup. And I was actually surprised when I started reading the literature that actually the prevalence thinking is that, no, they're not different. And as an example, I just want to quote you this review where the author said, well, the act of naming the first extending axon pioneers did not endow them with special properties lacking in other neurons. The ablation often perturbs the guidance of subsequent follower axons, either by delaying or misrouting them, but does not necessarily prevent followers from extending and locating their appropriate targets. And it was really interesting to me because they reference a couple of papers here, and I just read this paper prior to reading the review. My take was completely opposite on this. So this is what this paper, Pike et al., which is actually a study from Judith Eisenlab in zebrafish about pioneer motor neurons, describes. So in zebrafish, there are primary and secondary motor neurons in the spinal cord, and there are only 3 primary motor neurons that are born during first day of post-fertilization in zebrafish in every somite or muscle segment. And they innervate, as indicated here, different parts of the muscle in the trunk, okay? And a day later, about 20 to 40 secondary motor neurons are born, and they use axons of these neurons essentially as pioneers. So they fasciculate to navigate to their targets. Now, since they're so distinct, what these researchers did, they went in and they ablated middle or caudal motor neurons. So this is rostral, this is middle, this is caudal, okay? And then they look whether secondary motor neurons can navigate appropriately without pioneers. And here you're looking at ablation of the mid or middle motor neuron. Actually, so if you look at the top, I think it's dying. So if you look at the top, so this is ablation of CAP. And this is unablated, and it's staining for secondary motor neurons. So you see they extended down to the trunk, and these neurons begin to extend. They delayed. However, they will eventually extend. So in this particular case, CAP is not absolutely required for navigating of followers. However, this is completely in contrast to the case when you ablate mid. So this is unablated, and this is again unablated, and you can see they extended. In this particular case, vast majority of secondary motor neurons cannot extend, okay? So it seems to be actually case dependent, and it's true actually for other examples. If you look at the grasshopper, which has been a classical system for studying pioneer neurons, or you look in the xenopus or in zebrafish, in many cases it is required, but in some cases not absolutely required. So, which brings me to the question that we want to address, yes, it is required, but what I've told you so far, everything about pioneer motor neurons is actually about their morphology or their behavior. And surprisingly, we know little to nothing about molecular makeup of pioneer neurons and how they're different from the follower neurons. So we set up to essentially answer this question using zebrafish lateral line as a model system. So I just want you to tell you a little bit more detail about zebrafish lateral line. So the lateral line, both the sensory and neural domains shown here in green and magenta, derived from a structure called posterior lateral line placoid that is positioned just posterior to the developing zebrafish ear. At about 22 hours post-fertilization, posterior lateral line primordium, shown here in magenta, will migrate along the flank of the zebrafish. And as I mentioned, it will drop off these clusters of cells called neuromass that will develop into sensory organs. In the meantime, pioneer axon growth cones are embedded within this migrating population of cells, and sort of they extend all the way to the end of the tail, and they will innervate this posterior sensory organs, while follower axons will actually innervate more proximal sensory organs, and it will be important during later during my talk. And this is essentially a movie illustrating this process midway through the trunk, about 30 hours post-fertilization. So the way we got into thinking about pioneer axons and pioneer neurons is from the project that actually Katie started in the lab when she was a postdoc in the lab, and it's looking at the retrograde trafficking of activated neurotrophin receptor RET. What we noticed when we were looking where RET is expressed, that it was actually expressed in a subset of posterior lateral line cells. So here you're looking at transgenic line that labels cell bodies of all pioneer neurons in green, and here is a fluorescent in situ hybridization for RET message. You can see that it's upregulated in the dorsal neurons. And that was interesting to us because cell bodies of pioneers generally reside in the dorsal region of the ganglia. Okay, so that suggested to us that maybe these neurons are molecularly different. Now, another important question, which I already alluded to, are they actually required for the lateral line nerve extension? And we think yes, because when you look at the null mutant for the RET receptor, and you look at the lateral line nerve, so this is, again, the same transgenic, which marks many CNS and PNAS neurons, but you should be looking at this lateral line ganglion in 3D wild type sibling. So you can see the nerve extended all the way to the end of the tail. However, in the RET mutants, it extends only about 2/3 to 3/4 into the tail, and it almost never innervates the posterior sensory organs. However, the more proximal organs seem to be innervated normally. So that suggested to us that pioneer neurons are indeed required for the lateral line nerve extension. Now, how do we, and at this time, you know, when we were started thinking about this, the single cell RNA-seq was coming online. So of course we got on the, you know, single cell wagon. So the question, of course, first is, you know, how do you isolate lateral line neurons? About 20 to 30 neurons in the embryo at the time when these axons are extending, pioneer axons. So this is the transgenic line that I've been showing you, where regulatory elements of a proneural genes NeuroD driving cytoplasmic GFP. And not surprisingly, since it's proneural gene, it's expressed in most of the cranial ganglia. So posterior lateral line ganglion shown here. These are other posterior cranial ganglia, both the brain and the spinal cord, as well as, you know, olfactory epithelium, retina, and some other populations. So what we noted that it's actually expressed for some reason at higher levels in the cranial sensory ganglia. So we fact sorted these cells at 30 hours when the nerve was about halfway through the trunk. And we gated for only the brightest GFP cells to enrich for cranial sensory ganglia. So then, you know, we also probably seen these pictures before. So we did clustering of the cells, and each dot is a cell on this UMAP space. And we were able to assign, you know, cranial ganglia to this large cluster, brain and spinal cord, olfactory, epithelium, pancreas, and so forth. So now the question is, where are the lateral line neurons? Okay, so we know of many markers that are expressed in the lateral line neurons. So I'm showing you 4: RET itself, Stasmin, R-spondin, and the BMP molecule involving BMP signaling. So these are feature plots, which essentially show expression of the gene. If you like squint and stare at these for a while, you realize that they all expressed in the cluster that I outlined with this red circle. And this is these teal cells. I think they're teal because Lauren has a dog named Teal, and she likes teal color. She colored them teal, I'm pretty sure. And so these are lateral line neurons. And we can identify other subpopulations of cranial sensory neurons, like statoacoustic epibranchials. Now, of course, the most interesting question is now, is there any transcriptional heterogeneity with this population of lateral line neurons? So we can pluck out these cells, a couple hundred of these cells, and do unbiased subclustering. And these neurons break out into two clean subpopulations. I'm going to call them red and blue, or teal. And now the other question is, of course, where is red? And it turns out that red is upregulated in red cells, but not as high or not expressed in blue cells. In fact, if we do differential expression analysis between these two cell populations, there are about 30 plus genes that are differentially expressed. And in some cases, genes are almost exclusively expressed in red plus or red minus neurons, okay? So here's another way of looking at gene expression using dot plot, where the size of the circle corresponds to the percentage of cells that express a given gene and color to the level of expression. So on top are red positive cells and at the bottom red negative cells. And the first four genes are the ones I showed you on the previous slide. So all except red are equally expressed between two clusters, indicating these are lateral line neurons. And then we have genes that are have higher expression in red plus cells, and then we have genes that have higher expression in the red minus cells. So a couple pathways jumped out at us right away. So there seemed to be differential expression of different neurotrophins in these two populations of neurons. So of course, red and its co-receptor GFR alpha 1A expressed in red plus cells, as well as NTRK1. But NTRK3A is actually almost exclusively expressed in red minus population. So another interesting pathway, which I'm going to come back later to, is retinoic acid pathway. So CRAB-P1, and these are expressed in the follower cells at higher levels. So CRAB-P1B is a retinoic acid binding proteins, and these three genes are transcription factors that are known to be downstream of the retinoic acid pathway and transcriptional targets of the retinoic acid pathway, especially HUXB5A is kind of a bona fide target of the retinoic acid pathway. So that looks really good. So what we needed to do at this point is two things: confirm that our gene expression data are real in an animal, and two, actually show that red plus cells are indeed pioneer neurons. Okay, so in order to confirm our gene expression data, we did single molecule in situ hybridization using RNA-scope platform. And in this particular case, every dot presumably corresponds to a single RNA molecule. So what we did, we actually did pairwise in situs. We took either two red plus genes from red plus cluster, red minus cluster, or one gene from red plus and one gene from red minus cluster, and then calculated correlation coefficient. Okay, and in this particular case, we're actually using different transgenic. It's actually labels cell membranes of all posterior lateral line neurons. And the reason we did this because we can take it into linearis, create a surface for every neuron, and calculate number of actually dots. So that's how we quantify the data. And then we can, generate heat map like this, where we have three red genes from red plus cluster, three genes from red minus cluster. And what you can see that when you look at pairwise expression of genes from red plus, they highly correlate. And it's true also for red minus genes. But if you take gene from red plus and red minus cluster, they either do not correlate and anti-correlate. So this is really confirms the data that we see with our single cell. So the second question is, well, is red plus neurons are really pioneers? In order to address this, we knocked in mRuby protein into RPZ5 gene, and this is one of the genes that pretty much exclusively expressed in a red plus cluster. And as you can see here, again, cell membranes of lateral line neurons are labeled in green, and I outlined the ganglion. So you can see there is a subpopulation of mRuby positive cells in the dorsal aspect of the ganglion. And because it's a cytoplasmic fill, we can look at the primordium, and we always see that pioneer neurons are labeled. Okay, so this is actually confirms our hypothesis that our red plus cells indeed a population of pioneer neurons. Okay, so that was really interesting, and we have a lot of questions, and I think we can follow a lot of interesting leads based on the data we have. But the two main things we're asking right now is, where do they come from? In other words, what is the lineage of these two different populations of neurons? How did they separate during development? And what are the molecular cues, the driving specialization of pioneers versus follower neurons? Okay. So in order to tell you, in order for me to tell you about the lineage, I have to take you back to some classic embryology, okay? So as I mentioned, posterior lateral line neurons, as well as posterior lateral line primordium, derive from a posterior lateral line placoid. And that posterior lateral line placoid derived from a domain called panplacoidal ectoderm. So here is a schematized, vertebrate embryo. So the neural ectoderm is light blue and non-neural ectoderm is a dark blue. And between these two domains, sandwiched two other regions of ectoderm, neural crest, and panplacoidal ectoderm shown here in red. So panplacoidal ectoderm will give rise to all neurogenic and non-neurogenic placoids like lens, olfactory, epithelium, epibranchial ganglia, the ear, and the lateral line. Okay. So, and there are a number of genes that are specifically expressed in this horseshoe-like domain, including DLX, IS6. So here's a zebrafish embryo at 10 hours post-fertilization. So this is end of gastrulation and beginning of somatogenesis and neurulation. And you see this distinct horseshoe domain. So this arrow indicates sort of a thickening of the posterior aspect of this domain. Presumably, this is where otic placoid is coming from. And more posterior, presumably cells that give rise to the posterior lateral line. Just 4 hours later, if you look at the expression of neuro-D, and again, this is a proneural transcription factor, and now we're looking sideways, so this is the developing otic placoid. Just posterior to the otic placoid, you see this group of cells that begin to undergo neurogenesis. And again, just 4 hours later, so this is 18, which is in zebrafish 18 somite stage, the proneural cells already separated from the sensory cells that will give rise to the lateral line primordium. So again, in orange is in neuro-D, and in blue or, I don't know, purple, there is a staining for FGF10, which is expressed in the sensory domain. So things are happening pretty quickly, all right? And we stuck. There we go. So again, this is the same slide. I need to give you one more piece of information before I show you some data. So another piece of information is about how these two proneural and sensory cells separate from the posterior lateral line placoid. There was an interesting paper from Matoyoki Ida's lab about 10 years ago that actually showed through fate mapping that these cells presumably are intermixed within this posterior lateral line domain. And then somehow magically they separate, okay? So I'm telling you all this information because we really wanted to be able to trace posterior lateral line neuroblasts from the time they're born until they become isoprioneers and follower neurons, okay? So lucky for us, the transgenic line that we used to label cell membranes of posterior lateral line neurons also labels posterior lateral line placoid, okay? And we actually have a neuro-D-driven D-tomato transgenes that label cells that undergo neurogenesis. So here is the side view of the embryo at 15 hours post-fertilization. So this is onset of this transgene neuro-D-tomato expression about an hour after we can see the message by in situ. So here's the neural tube. Here are the forming otic placoid. Usually I go through stills and then just, let the movie play. But in this particular case, I'm just going to let the movie play, and then we're going to go through stills. Okay. So I'll just kind of let you observe what's happening here. So I'm just going to let the movie play again, so we can see how these two populations separate. And then primordium begins migrating. So basically, what I hope you can appreciate that the cells that undergo neurogenesis within the placoid initially indeed disperse throughout the placoid, maybe a little bit more toward the dorsal aspect of the placoid. Within a couple hours, they actually delaminate and migrate upward or dorsally and segregate into the ganglion, while the cells that are left behind form the prosensory domain that will become the lateral line primordium. And then at about 21, 22 hours, you already have a few axons of presumably pioneer neurons that invade this cell population, and then they will actually co-migrate with these cells and they sort of leave the scene, okay? So that was good because it actually told us about important time points that we need to be looking at, but it still doesn't tell us where the pioneer neurons. Although, when I was looking at this movie, first of all, I said, wow, this is really cool. But second is, well, I know where pioneers are. They probably cells that contact prosensory domain, and probably prosensory domain is talking to the neuroblast and telling them, become pioneer, for example. Of course, I was wrong. So we said, okay, we need to label pioneers so we can see what's happening. The problem is that our RPZ5 mRuby line took forever for mRuby to fold, so it just wasn't useful, and probably RPZ5 was expressed too late. So we generated another knock-in line that was a little bit more informative, and so we knocked in GFP into GFR alpha 1A locus. So this is co-receptor for Rett that is also almost exclusively expressed in pioneers. It was still too late, but it was a little bit informative because we can actually just start seeing GFP expression shown here with an arrow. And this is just GFP channel at 18 hours. And this is when these two sort of domains are closely opposed to each other, but not separated completely yet. I'm just going to play this. Maybe. So what I hope you can appreciate that, there is a subset of cells, and I should say that in this particular case, membranes, red membranes label the plaque code and cell bodies of neurons, as well as the sensory domain. So the one thing that, we were able to appreciate from this movie is that cell bodies of pioneers seem to be actually distributed kind of randomly and intermixed with what we think are follower neurons, right? So they're not necessarily all sitting, just at the distal aspect of this forming ganglion that touching the sensory domain. So we said, well, we really need to label these cells earlier. So we use transient expression assays where we can, with NeuroD promoter, drive M-cherry just in a few cells within this proneural domain. And we can label either pioneers or followers. So in this movie, there are two cells that are pioneers that are labeled, okay? And the movie starts at 17 hours post-fertilization. And what was immediately interesting, that first of all, if you look at the shape of these cells, they're spindle-like. And if you look where the actual neurites or protrusions of these cells, they're already invaded the sensory domain. So you kind of can see them here, okay? So they're in contact with the sensory cells. So you can see that they're sort of following or tracking the migrating cells, and the growth cones are within the actual population of these migrating prosensory cells that sort of leave the scene. But the cell bodies, they're not necessarily really located, rostrally or caudally. They kind of, at least as far as we can tell, within, randomly positioned within this population of lateral line neuroblasts. Okay. So that was slightly different from what we observed when we actually labeled follower neurons. So here are three cells, two bright and one dim, labeling the follower neurons. And right away, you can see it's at the same stage, same time point, that these cells are round, okay? And they are not contacting the sensory domain. And I hope you will see that their actually behavior is different. So they have this multipolar morphology, so they kind of extend neurites in all directions, and it goes for like 6 or 7 hours, okay? Until finally they will kind of get into the spindle shape form, and they will extend their growth cones. Although, we don't have a great sample yet, I think their growth cones are smaller. And what's also interesting, these growth cones are never invade this sensory domain. You can see they're like sort of trying to, but no, the train left the station. So they never actually co-migrate. So what we think is happening, potentially happening, is that there is some crosstalk between the sensory cells and the neuroblasts. And somehow this sort of specification into pioneer follower type happens pretty early, soon after neurogenesis of these cells. So then we were wondering, okay, so if that's happening, how does the gene expression changes during this critical time points? And now there are no questions that you cannot answer with a single cell. So we collected samples from 14 hours, presumably when neurogenesis begins, 18 hours, when these two domains are separate, but still touching each other, 22 hours, just before the sensory domain migration and active posterior lateral line neuron extension, and for good measure also at 48 hours, because we were curious whether the differences in pioneers and followers still persist at 48 hours. And I should disappoint you right away that 14-hour time points failed and actually being sequenced now as we speak, but there's some still interesting information from the rest. So I'm not going to show you the big clustering diagram, but suffices to say that when you're trying to look at different differentiation states, cells that go through differentiation process typically cluster next to each other during single cell clustering. So, we were hoping to identify a cluster next to pioneers and followers that will actually contain progenitors for the pioneers and followers, okay? And so there was this blue cluster that was next to pioneers and followers. And what was interesting that 14-hour time points contributed itself to this cluster, but not pioneers and followers, so it was a good thing, right? And more cells from 22 and only a few to pioneers and followers, more to followers, well, both. And also, so this is our old data, and at 48 hours, these differences in pioneer followers still persist, right? So this is the end of axon extension. So now we just needed to convince ourselves that these blue cells are indeed progenitors for pioneers and followers. So we can do it in a few ways. So we did a pseudotime analysis to show that these indeed are, older cell by pseudotime, and these are the youngest cells. So that was consistent with them being progenitors. So one thing I didn't tell you that we and others know that these neuroblasts are postmitotic, they don't divide. So we look at PCNA expression, and there's very little to no PCNA expression in this population. The earliest proneural transcription factor, neurogenin, is expressed in the cells. That's good, but not markers like SNAP25, SNAP25A and B, which are markers for more mature neurons. So at this point, we were convinced that we identified the right population. So then the next question we asked is, okay, so what is a pattern of expression of pioneer and follower genes over time? And that's where we actually were up for surprise. So if you look at expression of pioneer genes like GFR alpha 1A and RPZ5, they don't express in progenitors. They're only expressed in older cells, okay? However, if you look at the follower marker expression, it's expressed in both progenitors and more mature neurons at later time points. And this is actually even more obvious if we just isolate 14 and 22-hour cells. So the 14-hour cells are inbred, so differentiation trajectory is going this way. If you look at ZFHX3, which is a follower marker, almost all cells express ZFHX3. However, if you look at two pioneer markers, NGF receptor alpha and RPZ5, only most differentiated cells, very few cells actually express these pioneers. In fact, if we do a blend or look at the cells that contain expression of pioneers, they often also express followers, which we actually almost never see in the more mature neurons. So what this data basically suggested to us that cells, progenitor population born as a followers, and then through some type of mechanism, the pioneer marker expression is upregulated, okay? And at some point you see actually expression of both pioneers and followers, and then follower downregulated, and these cells presumably become bona fide pioneers. So can we verify this in the animal? So we did in situ hybridization at 30 hours and 20 hours to look at a pioneer and follower marker expression. So this is basically shows you expression of RET, which is pioneer, and ZFHX3, which is a follower. And you can see that essentially they're pretty much mutually exclusive. There are some cells that express ZFHX3 or RET. And when you plot expression like this, so this is basically like raw data, the way we analyze the data. So this is expression of RET, so it's number of puncta, and each dot is a cell. And here's a number of puncta of ZFHX3, and each dot again is a cell that expressing this number of puncta. You see that there are almost no to very few cells that co-express both. And we actually, in fact, when we go in and look, what these cells are, typically Imaris cannot separate two cells, and these are actually doublets, and it thinks it's one cell, but it's two cells. However, if we look at expression of RET as ZFHX3 at 20 hours, there are actually quite a few cells that co-express both. And again, RET is actually just coming up, so it's actually not expressed at a high level. So that's kind of what we think confirms our observation about followers being sort of a ground state, and then pioneer genes coming up. somewhere, between 18 and 22 hours post-polarization. So hopefully we'll resolve this when we have 18-hour time point. Now, of course, what we really want to know, like, what are the molecular cues that drive these gene expression changes and drive pioneer gene expression? And we have a model where we think there's possibly a crosstalk between the sensory domain and the neurogenic cells, and then the cells that actually invade the sensory domain receive some sort of a signal, and that allows them to differentiate. And we actually have an RNA-seq data sets from both, and you can do this analysis called NATME, where you designate posterior lateral line cells as a sender cell, and the neurons or neuronal progenitors are receiver cells, and it goes through all ligand receptor pairs that express in both populations, and you can prioritize them, and I'm happy to talk about how it does. So for example, if we look at our 14 to 22 hour progenitor population, so we designate the sensory cells, PLP cells, as a sender, and these neural progenitors as a receiver. So it will rank, and highlight the highest, ranked genes for you. And there are quite a few interesting receptor ligand pairs. So there are some cell surface molecules, Wnt and delta notch pathways. And basically what we're doing right now is in situ and doing transient CRISPR screen, trying to sort of go through in our RPZM Ruby transgenic to see if we can perturb number of pioneers and followers neurons. But, I don't have really good answer for you. However, I want to come back to the retinoic acid. So retinoic acid was actually one of the good candidate pathways for us that might regulate this balance between pioneers and followers, because one thing I did not mention, that work from Tatiana Petrovsky lab showed that you actually need retinoic acid to induce posterior lateral line plaque. But they never looked beyond onset of neurogenesis, but presumably it's still there. So we think there is active retinoic acid signaling within these cells. And then our hypothesis was, well, then you probably need to downregulate retinoic acid in order for these cells to become pioneers. So is there active retinoic acid signaling in the posterior lateral line neurons? So we use a retinoic acid sensor line from Josh Waxman lab, where retinoic acid binding domain from retinoic acid receptor is fused to DNA binding domain of GAL4. And when retinoic acid is present, this protein is able to bind upstream regulatory sequence and drive expression of GFP. So unfortunately, the line doesn't actually read very well low to medium levels of retinoic acid. So we have to fix these embryos and stain. And at 24 hours, you can actually see that after immunolabeling, there is a subset of cells within the lateral line ganglion, labels here with Hu antibody, that actually has GFP. So that kind of convinced that there is some retinoic acid signaling. So now, what we wanted to do is manipulate retinoic acid signaling in a single neurons within the ganglion. And in order to do it, we did transient injections of a plasmid that express either activated retinoic acid receptor or a dominant negative retinoic acid receptor in single neurons at the onset of neurogenesis. And then we looked at where their axons are targeted to assign pioneer or follower fate. So remember that pioneers will innervate the distal sensory organs, while follower will innervate more proximal sensory organs. Okay, so we can see these neurons because the construct has a cleavable peptide followed by EGFP CAX, and SIL is a lateral line specific enhancer. And as a control, we use just EGFP CAX. So if we analyze neurons, single neurons that are labeled with EGFP CAX within the ganglion, and we ask where they terminate, it seems to be fairly even, throughout the trunk. So L1 is the first neuromass, and this is terminal neuromass. There are typically about 5 neuromass throughout the trunk. Now, if we overstimulate retinoic acid, so it seems to be this is shifted to innervating more anterior sensory organs, and we never see innervation of the terminal sensory organs. So this is consistent with high retinoic acid levels that we see in the follower neurons. However, if we do opposite and we inactivate retinoic acid signaling, now we see this balance is shifting toward innervation of more terminal neuromass that are usually innervated by pioneer neurons. So this is consistent with pioneers having less retinoic acid signaling. Okay, so that was interesting. Now the question is, okay, if retinoic acid is regulating this choice, or at least targeting what are the molecules that it might be regulating? And if you remember, I already mentioned to you that RET is one of the neurotrophin receptors that is pretty specific to the pioneer neurons. And in the absence of RET, you don't innervate terminal sensory organs, okay? So we're like, well, we've got to look at the expression of RET when we manipulate retinoic acid. So, and as an internal control, we actually use HUX-B5A because it's a bona fide retinoic acid target. So, and this is just to remind you that it is expressed, although at not very high level, in a subset of the follower cells. And you can appreciate this on the single Z stack. So here's 2 dots in one cell, and probably a few cells have a few other dots, but you just cannot see them, okay? Now, if we look at DMSO expression, and we take a bunch of embryos and we count, a number of cells that express certain amount of HUX-B5A puncta. So what you see that, yes, the HUX-B5A is expressed at fairly low levels, 10 puncta or less in a certain population, and there are some cells that don't express HUX-B5A. If you inhibit retinoic acid signaling with DAB, so now you shift this median to the left. So now you have more cells that actually don't have any expression of HUX-B5A, and cells have fewer. But if you treat with retinoic acid, now you basically shift the median to the right. So there are really almost no cells that have no HUX-B5A expression, but there are cells with many more puncta. And this is sort of illustrated on these single Z stacks. Okay, so now what happens to RET? And what happens to RET is if you look at DMSO control, you have this bimodal distribution. So you have cells with high RET expression and cells with no or low RET expression. And presumably these are our pioneer and follower population. And when we do DAB treatment, we actually don't see median shifting very much. However, we do see that there are fewer cells that actually now do not express RET, right? So this kind of whole thing is actually shifted to the right, which kind of makes sense. And you can actually see it from this Z stack. There are actually more cells that actually have any kind of RET expression. The effect is actually more pronounced if we treat with retinoic acid. Now, basically what happens, this is shifted to the left, where we now have actually downregulation of RET expression throughout the RET ganglion, throughout the posterior lateral line ganglion, okay? So this is essentially telling us that potentially RET signaling is regulating, RA signaling, retinoic acid signaling is regulating RET expression. So we're doing more experiments now, sort of to verify that RET is really a target that regulating axon extension or axon targeting of pioneers. So to kind of wrap it up. So to summarize, what I told you that the posterior lateral line neuroblasts are essentially born as a follower population. Sort of they have this equal gene expression. And then through mechanisms we don't necessarily understand, but we would love to know, within a few hours, the pioneer gene expression comes up. And potentially this is downregulate retinoic acid in pioneers, and through this downregulation of retinoic acid in pioneers, we can express molecules like RET, which will target pioneers, to the distal sensory organs, but not more proximal sensory organs. Okay, so I'm going to stop here, and again, thank Ben, Lauren, and Nick, as well as other people in the lab, Katie and Molly, who actually started the RET project. People who gave us reagents as well as funding. And thank you for your attention. Happy to take any questions. Yes. Mary.
Hi, thanks, great talk. I was wondering when the diversity of primordium gets set up, like the different CFCR4 and CFCR7 domains, like they're, it's a pattern, right, in itself, and does that start at these really early stages, and could that have something to do with the crosstalk between the primordium cells and the sensory neurons to the pioneers going to the front part of the primordium and the follows in here?
So what Mary is referring to, when lateral line primordium is actively migrating throughout the trunk, there seems to be two molecularly distinct regions, the leading cells and the follower cells. And the gene expression, are very different. So I don't know the gene expression differences when primordium is actively migrating. But a few genes that we looked at, it's opposite actually. They start, the leading genes start at the back, at the caudal, at the rostral region, and then it sweeps through, okay? Now, what we're doing now, we actually sequencing sensory domain, doing single cell at 18 hours, because the data that I showed you actually at 24, when these differences are already set up that you mentioned. So we would like to sort of match, our cells at 18 neuroblasts to 18 cells, 18-hour sensory cells, sort of more precisely hold onto, these signaling molecules that we can profile. But we've tested some genes, and the obvious candidates for us was RET GDNF. So GDNF is expressed at that stage throughout the primordium at 18 hours, and RET, just beginning to be upregulated. And the neuroblast, we tested NTRK and NGF, and they're also both present. Again, it's specific for the lateral line. NGF is present in sensory domain, and NTRK is present in the neuroblast. So those were kind of our best candidates for now. But yeah, maybe it's good.
I was just wondering if you're in that set where you have the neurons all mixed together and they have codes, like they have indistinguishable expression pattern, and then you start separating. Have you tested, considered whether lateral inhibition by notch could be involved? Because that comes up over and over in similar situations, that at least in worms and flies, it's kind of the difference of that.
Yeah, for sure. So the question for people online was whether delta notch signaling and lateral inhibition that goes with it may be involved in separation of presumptive followers and pioneers. So we're actually doing it right now. So one interesting thing is that delta notch is regulating separation of the sensory and neural domain from the plateau. So that was actually, again, was in the paper I cited, the paper from 2011. But I don't know if you noticed when I showed the sort of data from this. So there is a jagged notch. So jagged is present in the sensory cells, and notch is present in our neuroblast, and there is also YBX and notch 1B. So definitely our candidates. And essentially what we need to do is quick and dirty experiment like DAPT treatment and see if we perturb this, but we haven't done that yet. For sure, yeah, that's definitely on my mind.
So you showed the effect of RET, the knockout, that there has some effect on the distal. But if you look sort of at the nice time lapse of the gene, is there any effect of that in the early extension of those processes? Is there anything you can resolve that's happening?
Yeah, so we don't know exact cellular basis for this defect where nerve would sort of extend about 2/3 and then just stops, and it just stops there. It's not that it's delayed, we can look many days later, it's still right there. So, but we did look at the growth cones, and it seems to be that initially when we look at the primordium, we see 3 to 6 pioneers. In the rat, we see initially 3 to 6. We look a couple of hours later, we see fewer, and a couple hours later, we see even fewer. And then finally, at some point, we don't see any pioneers. But the primordium will migrate even if there are no nerve associated with that. So the nerve will just extend up to a certain point and stops. And, I think we see fewer pioneers growth cones within that primordium. And I should say primordium secretes GDNF, so it's not surprising that they coupled. But how this dynamic of just, started with initially maybe normal number, and then it's kind of dropping off along the way.
But RET's on much earlier, though. Right? It's on the very initial stage of initial axon extension.
So RET is definitely right before axons begin extending, and probably coming up at about 18 hours when these two domains are just separated. Yeah. Does it answer your question, at least in part?
So I have one question about maybe differential attention. So it looks like when the cells separate and the pioneer neurons separate from the sensory, the leading sensory organs, right? It looks like there's differences in how well those cells are sticking together. Have you seen any, do you have any guesses on maybe differential attention that can play a role in the development of those pioneer neurons?
Yeah, so there are a couple of interesting things. So I don't know if they're sticking together, but they definitely look different. But they are getting sort of clumped together. And then at certain point, cells' bodies reorient themselves, right? Because initially they sort of intermix within the ganglion, and then pioneer cell bodies end up in the dorsal aspect of the ganglion. And I have no idea why that is. And I should say those cell bodies are bigger, for sure, than the follower cell bodies. And if we look at our single cell data, there is differential expression of protocoherence in pioneers and followers, but we haven't tested if it's significant. But there are some interesting protocoherence that is higher, not exclusive, but it's higher in lung population.
Any other questions for Alex? Great, thank you.
Thank you.